Development of a live attenuated vaccine for equine alpha herpesvirus 1. Equine alpha herpesvirus 1, EHV-1, poses a significant threat to the equine industry, causing respiratory diseases, abortion, and neurological disorders, including equine herpes virus myeloencephalopathy. The economic impact of EHV-1 outbreaks, particularly in the horse racing industry, underscores the urgent need for effective preventive measures. Current vaccination strategies using commercially available inactivated and modified live vaccines, MLVs, have shown limited success in controlling EHV-1 outbreaks. A promising approach to combat EHV-1 infection is the development of live attenuated vaccines LAVs that can elicit robust and long-lasting immune responses. LAVs stimulate both humoral and cellular immunity, providing superior protection compared to inactivated vaccines. However, developing LAVs for herpes viruses presents challenges, due to the virus's ability to establish latent infections and evade the immune system. One strategy for developing safe and effective LAVs involves deleting genes responsible for neuropathogenicity and immune evasion, targeting ORF37-38 for vaccine development. Previous studies have identified ORF38, a gene encoding a protein involved in viral DNA replication and transcription, as a key player in latent viral infection and virulence. Alpha herpesvirus uses with deletions or mutations in ORF38 have been shown to exhibit reduced virulence and an inability to establish latent infection. However, ORF38-deletion mutants of EHV-1 have been reported to induce adverse effects, raising concerns about their safety. The study focused on developing a double deletion mutant targeting both ORF37 and ORF38 to enhance safety and immunogenicity. ORF37, a gene belonging to the UL24 family of herpes viruses, has been linked to mouse encephalitis in EHV-1, although its exact role remains unclear. Construction and characterization of the ORF37-38 mutant using CRISPR-Cas9 gene editing technology. Researchers constructed the ORF37-38 double deletion mutant from a neuropathogenic EHV-1 strain, ORF30N752, named YM 2019, to ensure complete deletion. ORF37-38 was initially replaced with an EHV gene expression cassette, followed by the removal of the EHV cassette. In vitro characterization of the ORF37-38 mutant revealed the following. Replication. The ORF37-38 mutant exhibited herpes virus characteristic cytopathic effects CPEs in infected cells, confirming its ability to replicate plaque size. The ORF37-38 mutant produced slightly smaller plaques compared to the parental virus, suggesting a potential impact on viral spread growth kinetics. The ORF37-38 mutant exhibited a slightly lower peak titer compared to the parental virus in one-step growth curve analysis, but its overall growth kinetics remained comparable. Assessing neuropathogenicity and latency reactivation. The pathogenicity of the ORF37-38 mutant was evaluated in a Syrian hamster model. Acute infection. Hamsters infected with the ORF37-38 mutant did not display significant weight loss, clinical signs, or mortality, even at high doses. In contrast, hamsters infected with the parental virus at the same dose succumbed to infection within 8 days, exhibiting severe respiratory and neurological symptoms. Histopathology. Histopathological analysis of lung and brain tissues revealed minimal pathological damage in hamsters infected with the ORF37-38 mutant, further confirming its attenuated phenotype. Latency reactivation. To assess the mutant's ability to reactivate from latency, hamsters were infected with low doses of the ORF37-38 mutant and subsequently treated with dexamethasone, a potent inducer of herpes virus reactivation. Notably, the ORF37-38 mutant did not cause an increase in clinical signs, viral DNA loads, or pathological damage upon dexamethasone stimulation, indicating an inability to reactivate from latency. Evaluating immune efficacy and vaccine potential. To determine the vaccine potential of the ORF37-38 mutant, researchers assessed its immunogenicity and protective efficacy in hamsters. Humoral immunity. Intranasal immunization with the ORF37-38 mutant elicited high levels of EHV-1 specific neutralizing antibodies and IgG antibodies. The antibody response was significantly stronger compared to the single deletion ORF38 mutant cellular immunity. Immunization with the ORF37-38 mutant induced robust cellular immune responses, as evidenced by increased splenic lymphocyte proliferation and elevated levels of IF and Greek letter gamma and IL-10 cytokines. Protection against lethal challenge. Hamsters immunized with the ORF37-38 mutant were fully protected against lethal challenge with the parental virus, demonstrating its efficacy in conferring protective immunity. Even at a low immunization dose, the ORF37-38 mutant provided 100% protection, highlighting its potency. Advantages of the ORF37-38 mutant. As a vaccine candidate, the study presents compelling evidence supporting the potential of the ORF37-38 mutant as a live attenuated vaccine candidate for EHV-1. Enhanced safety. The double deletion of ORF37-38 significantly attenuated viral virulence, eliminating the risk of neuropathogenicity and reactivation from latency. This addresses concerns associated with the safety of single deletion ORF38 mutants. Robust immunogenicity. The ORF37-38 mutant induced strong humoral and cellular immune responses, superior to those elicited by the ORF38 mutant, resulting in complete protection against lethal challenge. Cost effectiveness. Despite a slight reduction in viral titers during in vitro propagation, the ORF37-38 mutant's growth kinetics remained comparable to the parental virus, suggesting no significant increase in production costs. Versatility. The ORF3738 mutant can serve as a platform for developing both traditional Latent herpes virus vector-based vaccines. Future directions. While the study provides promising results, further research is needed to fully translate the ORF3738 mutant into a viable vaccine for horses. Evaluation in horses, testing the safety, immunogenicity, and efficacy of the ORF3738 mutant in horses is crucial for clinical development mechanism of latency inhibition. Understanding the precise mechanisms by which the deletion of ORF3738 inhibits latency and reactivation would provide valuable insights for future vaccine design. Durability of immunity. Long-term monitoring of immunized horses is necessary to determine the duration of protection conferred by the ORF3738 mutant vaccine. Shedding of infectious virus. Assessing the shedding of infectious virus in vaccinated horses is essential to evaluate the potential for transmission. In conclusion, the development of the ORF3738 double deletion mutant represents a significant advancement in the quest for an effective EHV-1 vaccine. This mutant's enhanced safety, robust immunogenicity, and potential for versatile applications warrant further investigation for its clinical translation and contribution to the control of EHV-1 infection in horses.